Welcome to another video of Divya Mahesh tutorial. So in the last video we saw that Mishra discovered genetic material and as an acidic substance. So he named it as nuclein, and then we saw the functions of genetic material in genotypic, phenotypic and evolutionary ways. So today we will discuss about the genetic material, actually the DNA and RNA, their structure, their chemical structure. So when we'll come in DNA, so first we should know that DNA and RNA, they both are known as the nucleic acids, okay, because they are built with unit nucleotides, and those nucleotides are built with unit nucleosides plus phosphate, and those nucleosides are built up with the units of ribose sugar and nitrogenous bases. So in the structure of DNA, here it's DNA and RNA, difference lies at the two way. First, in DNA there is a deoxyribose sugar, and in RNA it is ribose sugar. So difference lies as deoxy means the absence of oxygen at 2' position in the sugar. So one, two, three, 4, 5. Actually it will be 1, 2, 3, 4 and 5 like this. So here we'll see that in this position, this second position here, OH is present in RNA, and this O makes RNA more reactive than DNA, sorry. So in DNA there is the absence of O in the 2' position. If RNA becomes more reactive, then it becomes unstable, we know. And also because of this, it acts as catalytic and adapter molecule. It is easy to act as catalytic and adaptive molecule, but DNA being more stable is easy to transfer from one generation to other. So in further videos we'll see why DNA is mostly the genetic material in almost organisms, because of its stability and deoxyribose sugar. So we saw this. Now the second difference lies in nitrogenous bases. These are nitrogen bases. So second difference lies in nitrogenous bases. In DNA there is a presence of T, and in RNA T replaced by U. Rest A, G, C are common. So in DNA there is a presence of thymine, and in RNA there is a presence of uracil. Now if we study the DNA and RNA, so we know DNA, a very good model, we can say model was proposed by Watson and Crick, double helix model of DNA. So in this they told that DNA is double helix. So what does double helix means? That two chains run anti-parallel to each other. So how they run, we'll see at molecular level first. So in molecular level we'll see there are one nucleotide chain. If we take, so polynucleotide chain, so we'll see pentose sugar. Pentose sugar is linked with nitrogenous base with glycosidic linkage, and this sugar at this position is linked with phosphate by phosphoester linkage, and thus this is a complete nucleotide. This is a complete nucleotide. And the same with opposite base pairing, like if we say here it is A, so A will pair with T, so here H in there, CH2O phosphate, here also CH2O, you can add CH2O phosphate, another one. So this is a complete nucleotide, and like this anti-parallel, so here it's 5' position and here it's 3' position, here it's 3' position. So one chain runs at 5' and another runs at 3'. This is anti-parallel, both the chains run, so they take turns, form the double helix. So like this more nucleotides will get joined, and there between two nitrogenous bases there is a presence of hydrogen bonding. So we have to learn that between A and T there is a double hydrogen bonding, and between G and C there is a triple hydrogen bonding. So this is the structure of DNA, and same is the structure of RNA, but the difference lies here with the replacement of this, and many RNAs are single stranded. So we studied the structure of DNA and RNA. Now double helix, if we focus on the double helix model proposed by Watson and Crick, double helix model for DNA, we'll see here that DNA is double helix. These are hydrogen bonds between nitrogenous bases, and this is a phosphate sugar backbone, the phosphate sugar backbone. So in DNA double helix, we'll see there are major groove and this is minor groove. This is one chain and this is another chain which take turns. Either it could be right-handed or it could be left-handed turn. So we'll see in the different forms of DNA, we'll see which DNAs are right-handed and which are left-handed. So we'll see there are nucleotides, base pairs in each turn. So in average we take bDNA, we'll see later, as a majority. So in that we'll see each turn has 10 base pairs. In each turn there is a 10 base present. So like this. And when we'll see the distance between two nucleotide base pairs, will get 3.34 nanometer, and this one turn distance 3.4 nanometer. So this is the double helix model which was proposed by Watson and Crick. So this is basic. We can learn from book also these. So we saw that nuclein was proposed, we saw the functions of genetic material. Now we discussed about the structure of DNA and RNA. So we got the total knowledge of structures of DNA and RNA. Now as the scientists got these structures and all, then they started doing their experiments on whether the DNA acts as a genetic material or RNA acts as a genetic material. In which organism, किन organisms में as DNA act करता है as a genetic material और किस में RNA act करता है as a genetic material. So various experiments were proposed. So those experiments we will see in the later classes, and we'll also see different forms of DNA, different types of DNA which are found in the organism. So I end my video. Thank you.